Human gene therapy seeks to modify or manipulate the expression of a gene or to alter the biological properties of living cells for therapeutic use. It is also a technique that modifies a person's genes to treat or cure diseases. And today in this vlog, we will tackle more about gene therapy by answering some of the guide questions provided by Group 5.
Gene therapy involves altering the genes inside your body cells in an effort to treat or stop diseases. Genes contain your DNA, the code that controls much of your body's form and function, from making you grow taller to regulating your body systems. Genes that don't work properly can cause diseases. Genes may be introduced, removed, or changed in the cells of patients receiving gene therapy. This mostly experimental technique changes how the cell produces proteins. On the other hand, cell-based therapy involves modifying a patient's own cells or cells from a donor to fight disease and alleviate medical conditions. Cellular therapy is the use of viable human cells transferred into a patient to replace or repair damaged tissue or cells. Blood transfusion is an example, and stem cell therapy is just one of the innovative techniques used in a variety of diseases.
In order to deliver the genes into the nucleus of the host cell, a vector is needed. If genes are directly introduced into the cell, they do not work. The vector then infects the surrounding cells in the tissue, effectively delivering the genes vectored by the viruses. Alternatively, the affected cells are removed and infected in the laboratory with viruses with engineered genes. However, there are many challenges in this technique. Firstly, this technique is largely experimental and therefore far from perfection. Secondly, they have to make sure that the newly introduced gene actually works by synthesizing the desired protein. In simple terms, gene therapy works by replacing a defective gene, changing the genetic information of a population cells for the purpose of the prevention of a disorder or alleviating symptoms of a disorder.
Well, there are 4 strategies that can be used in gene therapy. Firstly, we have gene replacement that can be used for monogenic diseases. And second, we have gene addition, which is for complex disorders and for other infectious diseases. And next, we have gene expression alteration targeting RNA. And lastly, we have gene editing to help introduce targeted changes in host genome.
Gene therapy replaces missing or defective genes, deliver genes that speed up the destruction of cancer cells, supply genes that cause cancer cells to revert back to normal cells, deliver bacterial or viral genes as a form of vaccination, provide genes that promote or impede the growth of new tissue, and also deliver genes that stimulate the healing of the damaged tissue.
The most common type of cell therapy is blood transfusion and the transfusion of red blood cells, white blood cells, and platelets from a donor. Another common cell therapy is the transplantation of hematopoietic stem cells to create bone marrow, which has been performed for over 40 years. With gene therapy, cell therapy subtypes can be classified in different ways. There is currently no formal classification system for cell therapy. Here, the different types of cells used for cell therapy have been classified by cell potency.
Viruses are used in gene therapy as delivery vectors and as oncologic viruses. Viruses as gene delivery vectors are tasked to deliver the genes to the cell. and only genetically engineered vectors such as viruses are used in gene therapy. This newly engineered virus protects the new gene it carries from degrading enzymes in the blood. The virus is an obligate intercellular organism that forces the cell to take up the new gene and go into the nucleus. Once in the nucleus, or for someone, once integrated into the chromosome, the new gene starts to perform its function to synthesize a new improved and functional protein. Furthermore, these viral vectors are designed such that their ability to replicate uncontrollably inside cells is removed. However, there must a room for a new gene. Oncolotic viruses. These viruses are engineered such that they replicate only in cancer cells, or predominantly cells causing the cancer cells to lyse. Released viruses during the cell lysis will now infect other cancerous cells, but not normal cells. Thus, these types of viruses are known as oncolotic.
No, because these viruses used in gene therapy are designed to reduce the risk of adverse effects. These viral vectors are tested in animals and in human trials to make sure that human cells are not harmed. However, like all treatments or therapy, it has its risk. Therefore, anyone availing of this therapy must discuss with their physician and other health advocates the pros and cons of gene therapy.